The overall goal of this video is to show researchers in the field of epigenetic study how to use the program GeNemo to obtain regions of similarity among different epigenetic marks and different cell types.
Our goal in creating this program was to enable researchers to compare their dataset of interest to the vast known public databases, such as ENCODE and MouseInc. In conventional search, you typically search by keywords, like ring, ES cells, H3K4 trimethylation, or other specific terms. While these methods are useful, GeNemo can also search for patterns in the actual data rather than similarities in the data description. Suppose you have a novel DNA binding protein with no or little a priori knowledge about what kind of epigenetic modifications or cell types you're expecting it to relate to. GeNemo can still help to narrow down the potential targets. GeNemo can read files in a variety of formats, including BAT and BigRead, and will use different pattern matching methods correspondingly. It will also sort the output regions based on their similarity score and provides data exporting options. And now we are going to show how to perform a GeNemo search.
To use GeNemo, you will need your own data file in BED or BigWig format, which can be converted from experimental raw data with common bioinformatics tools, such as the UCSC Genome Browser. First, go to genemo.org. On the main page, you will see a search box with specification options. Choose which species to compare your data with. For this tutorial, we will use Homo sapiens as the reference species. Then, choose your own file to upload to GeNemo.